Today we are going to study viroids. Viroids are they are not a virus. They were first discovered by the plant pathologist Theodore O'Diner in 1971. It is not a virus. Remember that it is not a virus. So basically, it was discovered by the plant pathologist Theodore O'Diner, and viroid is basically the smallest pathogen. You can say it is a piece of RNA in a circular form. What does it mean? As we know that virus has a capsid, and in its capsid virus, we are talking about virus, that virus has a capsid, and inside its capsid, that is RNA, is present. So that separate circular form RNA piece is known as viroid, but this complete is basically a virus. But that separate RNA circular form, that is known as RNA piece, and RNA piece is basically a viroid. Viroids basically have a non-coding region. A non-coding means it replicates, but it does not make protein. It replicates, their strands replicate, but it does not make any kind of protein. And it is a single stranded RNA with some double. This RNA, means viroid is basically a single stranded RNA with some double stranded region. Viroid was first discovered by the plant pathologist Theodore O'Diner in 1971, and it was first identified in the potato. This viroid, that is basically first identified in the potato, and it causes a disease. It causes a disease in potato, that is potato spindle tuber. Means in potato spindle tuber it causes a disease. So viroid mainly cause a disease in plant. But the hepatitis D, which in human hepatitis D can attack. So hepatitis D is basically considered as a viroid, but hepatitis D can only attack when hepatitis B is present in the person's body. Hepatitis B is present in the person's body. So hepatitis D in the human, if you talk about human, so hepatitis D is considered as a viroid, but viroid mainly cause a disease in plants. Till now, 33 species of viroid has been discovered. 33 viroid species has been discovered till now. They replicate, as I said, they replicate but does not make a protein. the viroid produces a specific RNA molecule. Okay, but it does not make a protein. There is no vaccine of it, and it is transmitted through if you talk about plant, because it mainly causes a disease in plant, so it is transmitted through pollens and seeds. If you talk about hepatitis D, so it can attack when hepatitis B is present in person's body. So it is transmitted through the pollens and seeds, and it stops the growth. You can see stops a growth. So now let's talk about that how viroid enters into the plant. Now, when viroid enters into the plant's body, basically it enters through the plasmodesmata. Plasmodesmata is basically a channel between two cells. Means it links or it connects two cells. So plasmodesmata is basically a channel. So through this channel, the viroid enters into the plant. And when it enters into the plant, it goes into the chloroplast, and when it goes into the chloroplast, the viroid affect the plant, in which the leaf gets yellow. When it attacks the chloroplast, the leaves get yellow, and why the leaves get yellow? Because the chlorophyll in the chloroplast ends, because of the viroid attacks into the chloroplast, and the leaf gets yellow because the chlorophyll ends on that chloroplast. And if it goes into the nucleus, enzyme is present in the nucleus that is known as RNA dependent RNA polymerase. This enzyme basically will make a strand but will not make a protein. Obviously the viroid did not make a protein, so this enzyme will make a protein, it will make a new strand. Then it replicates, and when the replication ends, then there's the enzyme that is known as ligase. Ligase enzyme is an enzyme that joins that viroids after the replication. This ligase enzyme will join that viroids, means these strands, it will join that viroid. So that is again repeating that when viroid enters, and basically it enters through plasmodesmata, that is a channel between the two cells. And if it enters into the chloroplast, so it affects the plant and leaf gets yellow. And when the leaf gets yellow, why it gets yellow? Because the chlorophyll ends into the chloroplast. Now when it goes into the nucleus, the enzyme is present, that is RNA dependent RNA polymerase. That enzyme basically make a new strand, and then after replication there is an enzyme that is ligase enzyme that join that viroids after the replication. Okay, when the RNA of viroid attacks the RNA of plant, means attach, when RNA of viroid attach with RNA of plant, viroid RNA will silence the RNA of plant. That is known as RNA silencing. Basically it does not make a protein, so how it can cause a disease? So that is when RNA of viroid enters into the RNA of plant, so the viroid RNA silence the RNA of plant, so that is known as RNA silencing, and it causes a disease.